Bismillah ar-Rahman ar-Rahim, my dear students of second year College of Pharmacy Pharmaceutics course. Today we're gonna start a new topic. This topic is the solid oral modified release dosage form and drug delivery systems. Now you will see from the title that we are focusing on solid dosage form. It's for oral delivery. The route of administration is oral. This is the focus. And we are talking about dosage form and drug delivery systems. And I already explained to you before the difference between a delivery system and a dosage form. Now what you should be able to answer by the end of this topic, you should tell me about the various types of modified release dosage form. So what are these types? When I say modified release dosage form, what are the types that belong to this category of products? You should compare and tell me the advantages and disadvantages of those modified release dosage forms. Generally, طبعا, you will tell me about these advantages, what are the good things, what are the bad things. Also, you should tell me what physical and chemical characteristics of the drug that allow the drug to be incorporated into such extended release dosage form. Also, you need to describe or explain what do you mean by microencapsulation, embedding a drug in a matrix, ion exchange, osmotic pump. You should explain them in relation to how we can achieve the modified release dosage form. Also, you need to tell me about the criteria that are used in the development of in vitro in vivo correlation. We say IVIVC, in vitro in vivo correlations for such extended release products. Okay, so these are the things you should know by the end of this topic. We are going to start with why, the reasons. When we say rationale is the reason behind something, why we doing it. So why do we need to do extended release pharmaceutical preparation? Of course, one of the first point anyone can say is taking multiple doses daily is inconvenient for the patient. And of course, there are many things or there are several consequences of this. Okay, if you have to take, for example, 3 or 4 times a day for any particular drug, there is a consequences of doing this. It's not only when I say inconvenient. No, you might be incompliant, you might miss dose, etc. Okay, so it's inconvenient, but the FDA says that we should not make modified drug release just because it's inconvenient. There should be other therapeutic benefits. Number one, it's inconvenient for the patient to take multiple doses every day. Also, when we do a comparison between a drug that is taken more than 1 time a day compared to a drug that is taken maybe 1 time a day, like what you see in this graph. Let's look carefully into this graph and try to explain it. You will see this is the time. The time means the time of taking the drug. Okay, this is 0 time, like for example you are taking it right now, you're starting with the drug. Okay, and every certain period you are taking the drug, as you see here. So you are taking your first dose here at 0 time, maybe after 8 hours you are taking your second dose, and another 8 hours you are taking your third dose, and so on. طيب So this is the time, and here is the concentration of the drug in your plasma, in your body. Okay, what you see here is two concentrations. Okay, this dotted line, you have two concentration. Now the drug concentration needs to be inside these two lines. What are these two lines? One represents the minimum effective concentration. So what it means that the drug must be above this concentration, or at least at this concentration, in order to produce an effect, a therapeutic effect. Okay, so if it is below this concentration here, the one that you see here, if it is below this, what is going to happen? The drug will be ineffective. Now the dotted line here shows the maximum safe concentration. Sometimes they call it minimum toxic concentration, MTC. Okay, this one is MEC and this MTC, minimum effective, minimum toxic concentration, which means the drug will start giving side effects at this concentration or above. So essentially, what does this tell you? That the drug concentration, you know, after taking the medication, should be in between these two lines, minimum effective concentration and the minimum toxic concentration. Okay, so the drug concentration should be between this line and this line. So anything here is good. طيب, what you see with multiple dose daily, you will see a lot of fluctuation. Fluctuation means going up and down. So here the drug starts, of course, at below the minimum effective concentration, because you're just starting with the medication. It gets absorbed from the GIT and into the blood. That's why the concentration increases and then reaches a peak. This is, we call it peak, and we call this one, we call it valley or trough concentration. So between peak and trough concentration. So start fluctuating between these two concentrations. So you have many fluctuation, and these fluctuations, especially if you don't stick with the time, with the correct time, most probably you will have concentration above the maximum safe concentration, or in another word, minimum toxic concentration. It's above that, and you have period of times, like from here to here, you have period of times where the effect is not seen in the patient, or there is no therapeutic effect. So instead, it's better to maybe take an extended release dosage form, like what you see in this dotted curve, okay, where you will see that you don't have such fluctuation and you can maintain therapeutic effect for a longer period of time. And of course, this is will be beneficial, especially if we are talking about treating chronic conditions. Yani you are not just taking it for a headache 1 or 2 days and that's it. No, but you are going to continuously benefit from the sustained or extended release dosage form. Okay. طيب, إذن immediate release dosage form has many peaks and valleys, and this might not give you the optimum therapeutic effect. طيب, what is the other thing which you can benefit from extended release is that you will eliminate the need for night dosing. Yani, let's say you have to give the drug every 4 hours, for example. This is just an example. It means probably at 2 o'clock you have to get up, okay, and you have to take the medication, or maybe at 12 o'clock, midnight, when you usually sleep, let's say, for example, at 9 o'clock. So this is, of course, again, not convenient, but it can be also troublesome if a caregiver, like a nurse, she needs to come and give you this medication because it needs to be given as multiple dose daily. So imagine how much you can have convenience if this is, you know, a single dose daily. Also, as we explained here in the graph, okay, you have an optimum concentration for long time, and you are reducing the possibility of side effects. فأكيد this is something good, is a reduction in adverse effects. Now, reduction in overall healthcare cost. You have to be careful, because here we are saying overall. Because if you want to compare the price of extended release dosage form compared to immediate release, لأنه عكس extended immediate. Okay, so if you do that comparison, actually the extended release dosage form is more expensive. But we don't look only at the price of this product. You have to look at the overall healthcare cost saving. Okay, because if you use the extended release dosage form, you're gonna have enhanced therapeutic effect. Okay, you're gonna have fewer side effects, is a better therapeutic effect, and fewer side effects, and also reduction in the time spent by the nurse or healthcare personnel to dispense and administer the drug, and also to monitor the patient. All these are reduced. That's why we say overall. Okay. طيب, another thing, and maybe this needs a little bit of explanation. Maybe you are aware that when these big pharmaceutical companies, when they start or they have a new drug into the market, usually, they need to file for patency, okay, even before the drug gets to the market. Okay, so when it gets to the market, usually 20 or less years will be left for the drug in the market, and after that the patency expires. Okay, and this allows other companies to come and benefit from this drug and produce it as generic drug product. Generic drug product will be sold at a much less price compared to the original company, the big pharmaceutical company that started this product, and they were the one who invented the product and spent so much money on the product. So compared to this, the generic drug product will be much less expensive, because these other pharmaceutical companies that want to imitate the original company, they don't spend as much money as the original company did. Okay, so by a little fraction of that cost the original company spent, the new pharmaceutical companies will be able to develop their product. So what happens is, okay, before the patency expire, the original company will start ways in order to extend the patency life. Okay, you know what I mean? It wants to say that, well, now we have new formulation, so you cannot imitate our new formulation, which is the extended release formulation. You cannot imitate, you cannot do extended release formulation. We have patency now, another 20 years for our formulation. So doing this will extend their patency into a new formulation. So they are using the drug for a new formulation, and nobody can imitate that formulation. So it's a way to profit from their new product using their old drug. Okay, طيب, of course you see that there are many nice things, but there are also some disadvantages of preparing or modifying the release of the drug. One of them is the loss of flexibility. I cannot go and break the tablet, okay, and say, okay, I want to break them into two halves and then I will take each half. Or I say, okay, I want to prepare it into a solution, so let me crush it and suspend it or dissolve it. No, we cannot do that. So there is a loss of flexibility. Yani if you compare this to immediate release dosage form, for immediate release dosage form, most probably you'll be able to crush it and dissolve it, for example, or suspend it in a liquid, okay? But you cannot do this with the modified release dosage form. Another problem is what we call dose dumping. Dose dumping, which means, suppose you are taking a tablet with a coat, and for a reason, maybe the coat was not proper or something else. Something else means the patient is taking, for example, a certain material like alcohol or something like that, and this results in a failure in the coating, okay, and then suddenly you have all of the drug. And remember, when I say all of the drug, for extended release dosage form, you have usually much more drug compared to immediate release. يكون كمية, the quantity of the drug is much more, because you want to give the drug for a longer period of time compared to immediate release. So you need to put more drug. So more drug is released suddenly. So what can this cause? It can cause it can cause toxic effects or side effects. Another thing which I already explained is that it's more costly compared to immediate release dosage form, because you need to do a lot of works on development, okay? Okay, now there are so many terms that are used, okay? Maybe they want to hint to something similar, but they are not the same. You have to be careful, because each type can have a different feature, okay? For example, you might say sustained release, sustained action, prolonged action, controlled release, extended release, time release, long acting, okay? And all of these terms have been used to describe drug products with certain features. For us, we need to know that usually the drug release features are based, it's based on what? It's either based on time, like after 1 hour, 2 hours, or 3 hours, the drug will start releasing. So it's based on time, or based on its movement, its course, we call it. So it's based on time or based on movement. For example, it goes to acidic, to the acidic stomach, then to the less acidic intestine, etc. Or to the location, the place, certain specific place, for example, the duodenum or the small intestine, okay? So the drug release features are based on these three things. And the United States Pharmacopoeia, and this is very important, differentiates or classifies the modified release forms into two classes, okay? One اللي هو extended release, and the other one is the delayed release. Again, the United States Pharmacopoeia differentiates or classifies modified release forms into two categories. One, extended release, and two, اللي هو delayed release forms. طيب, extended release, what does it mean? According to FDA, extended release allows a reduction in dosing frequencies, as you know, as compared by immediate release dosage form. So you have less dosing frequency. Instead, for example, of 4 times a day, maybe it's 2 times a day or 1 time a day. Okay. Delayed release, it's very important to differentiate it from extended release. Delayed release means the release is not promptly, not as in the immediate release dosage form. It's not promptly. Promptly yani immediately after swallowing the release will start. No. Delayed release معناته this will not happen as soon as the drug is administered, but there will be a delay, and this delay can be based on time. Okay, remember here we said that it can be based on time, okay, or the environment اللي هو like the pH اللي هو location, the pH اللي هو location. Okay, so the delayed release طبعا is like the enteric coated tablets, and you have studied this before. طيب, the other term that you need to be aware of اللي هو repeat action. Repeat action. In repeat action, usually we are talking about a tablet that may have 2 layers, or it's a tablet within tablet, and we have studied this before in the types of tablets. We have studied this. طيب, in this tablet you have you have 2 doses, 2 single doses. Okay, one is for immediate release, يعني as soon as you swallow it you start to have or the drug will start to be released, while the second one gets released after some time. Now it gets released after sometimes as يعني after, let's say, 4 hours starts releasing مثلا. Now this happens either as a dose, immediate release dosage form after 4 hours مثلا, for example, or as extended release tablet. Okay, so as extended release or as immediate release. Okay, that is after the release of the drug from the first layer. So the second layer starts later. Amma when we say targeted release, here we are talking about concentrating the drug in a specific place or region. Okay, why we are concentrating the drug at that area? Maybe because that area is where the drug will exert its action, or because it's the best place for absorbing the drug. Like we know that the small intestine is the best place for absorbing the drug. Okay, now we will talk about the extended release oral dosage forms, and we will give you examples. Before we give you examples, we need to talk about what type of drug that is suited for or to be in extended release formulation. What is this characteristics? Type the number one characteristic, it should not be absorbed or excreted or eliminated يعني, from the body, or absorbed into the body. This should not be very fast or very slow. If it is very fast or very slow, this will not work. Why? If this happens very slow, absorption or excretion, it's very slow, it means that the drug is already sustained release. It's already sustained. We cannot formulate it into, it's already having that characteristic. We cannot say, for example, okay, we'll give the drug every 1 month once, okay, orally. Okay, that's not the right thing. And we know that, things in our belly or in our GIT will remain, let's say, for 1 day, okay, or couple of days, but not more than that. Type. What about if it is very fast? If it is very fast, it means that I will need a big dose to be able to carry or to compensate what is lost. Okay, if the absorption or excretion is very fast, so I need a large dose to keep putting the drug in the system. So how can I put large dose as an oral solid oral form? So it should not be again, the drug should not be absorbed or excreted or eliminated very fast or very slow. It also should be uniformly absorbed across the GIT. Yani, for example, if I tell you, oh, this drug can only be absorbed in the stomach, okay, and we know that, you know, the drugs will not stay in the stomach for more than 2 hours. معناته I only have a window of 2 hours, صح? But if I tell you that the drug can be absorbed in the stomach, in the small intestine, and in the large intestine, and the absorption will be uniform, يعني similar, then we'll say, oh, okay, so I have almost the whole day for the drug to be absorbed in the GIT. So I can say, okay, I'm gonna formulate it as sustained or extended release product. Another thing which should be taken into account, that the drug dose should be small. Again, because if it is not small, if it is big, then you know that in the extended release of dosage form, I need to maybe double or triple the dose. For the extended release, I have to put more than one dose. So they have to be small so I can put them together. Also, they should have good margin of safety. Remember the graph that I showed you earlier? Okay, if we go back. Okay, this is the margin, safety margin, اللي هو the difference between the maximum safe concentration and minimum effective concentration. So this should be wide. So if there is any failure, لو صار أي failure في التكنولوجي, it will not cause a big problem. Okay. Also, it's preferred that it's given for chronic condition because you will benefit. Okay, if it is only for one day, you are having headache today and you are taking it just for this day, then you might not benefit a lot from, you know, having this extended release dosage form. Okay. طيب, how can we achieve? What are the technologies that can allow us to achieve what we talked about before, اللي هو based on time, based on course, based on location? What are the technologies available? اللي هو, one, the use of a barrier, coating barrier. يعني we coat the drug. So the fluid does not have access, body fluid, okay, does not have access to the drug immediately or easy. Another thing is to control the drug diffusion. Drug diffusion يعني going from the tablet, okay, this is the drug here, and the drug goes to the surrounding from the tablet. So how it goes, we control the rate of diffusion, how fast it diffuses from the tablet to the surrounding. Also, we control or we use the chemical reaction with specific site. Okay, the chemical reaction can be pH. Okay, the pH, for example, acidic pH will react with basic drug, for example, or less acidic or basic medium will react with acidic drug. So we control the chemical reaction between the drug or its barrier and the biological fluid. So these are the technologies that we are using. So what are the examples? So now we're gonna talk about examples. The first example, what we call it, اللي هو coated beads granules and microspheres. Okay, beads يعني خرزات. Okay, it's similar to spheres. Okay, when we say microspheres, first we are talking about spherical things, and they are in micrometer, يعني small. Okay, but let's see what we are talking about here. First, when we are talking about beads or pellets, خرزات برضو, pellets, or even granules, in order to prepare that, اللي هو coated beads, what we do is we put them in a conventional pan, and you have taken this in tablet coating, conventional pan, or the other way اللي هو air suspension coating, and we have taken this also in coating. Okay. طيب, so what do we do with the drug? The drug is sprayed, يعني the drug is dissolved in solution, and then it's sprayed on the beads. Beads are in the conventional pan or air suspension pan or coating, sorry. Now remember, if you remember, we put the tablets for sugar coating, we put the tablets في pan, and the pan starts rotating, and then we spray the sugar on the tablets. Now we do similar thing here, but instead of tablets we are either using nonpareil seeds. Nonpareil seeds, طبعا nonpareil here indicates sugar and starch, يكون sugar and starch. Okay, يعني particles like that مكون من sugar and starch, and then we spray on them the drug solution على coating pan. Okay, and this will cause them to be coated, and it will cause them to release the drug in a specific way. Now the nonpareil seeds, the coated one, will be in the size of 425 micrometer to 850 micrometer, which means less than one micrometer. And the other way, instead of coating the sugar and starch, we can coat the microcrystalline, and this one is preferable. Okay, it gives us a size from 170 to 600 micrometer. Again, it's not very far from the nonpareil seeds, but the good thing about the microcrystalline is that it's more durable during production. Okay, يعني we can produce these spheres. Okay, هاي الspheres اللي هي من المicrocrystalline easier compared to the production of nonpareil seeds. Okay. طيب, we can do it also with granules. Okay, so we can form granules, and you remember how granules are formed. If the drug needs to be in a large dose, then we cannot just coat granules and nجيب granules من excipients and coat them. We cannot only do that. We have to start with the drug and make it as granules from the drug, starting from the drug itself. طيب, then now I have granules made of what? Granules made of drug. Okay, now I will keep some of them uncoated. Okay, highly granules for the drug with large dose. Some of them I will keep them uncoated, and some of them, maybe 2/3 to 3/4, I will apply to them varying coats. Okay, يعني portion of them I will apply a lot of coats, some of them apply less coats, so varying amounts of coats or number of coats with materials like beeswax, carnauba wax, وهاي الماتيريال that you see here like glyceryl monosterate, cetyl alcohol, ethyl cellulose. This material actually will retard. It will not allow these granules to dissolve easily, so they will take longer time. So when you make them into tablets, you compress the granules. أي granules? The uncoated one with the granules that have been coated with varying degrees of coats. When I put them together, mix them in طبعا certain proportion for each one. When I put them together into tablets, or I put them in a capsule, what's gonna happen? The uncoated granules will dissolve first, and then materials that have been, or granules that have been coated with less beeswax, مثلا, for example, will dissolve faster than the one with a higher degree of coating, okay, and so on. So I will have like extended release form. Okay, so this is one example that we talked about. Now the second example of extended release is referred to as multi-tablet system. Multi-tablet system. Here we are talking about compressed tablets. But these compressed tablets are small spheroids. شو يعني small spheroids؟ يعني they are more spherical, okay? And they are very small. They are not like normal tablets, okay? Normal tablets will be 12 millimeter, 15 millimeter, and so on, 10 millimeter. But this is 3 to 4 millimeter. That's why they call them mini tablets. طيب, what do you do with this? Now, some of these tablets will be coated, okay, and some of them طبعا the coating applied will be يعني maybe not the same coating, يعني the thickness of the coat will be different that will be applied to them, to these mini tablets. Some of them will not be coated, some of them will be coated lightly, some of them will be coated more. And then you fill about 8 to 10 of these mini tablets, you fill it in gelatin capsule, so that those uncoated mini tablets will provide immediate release, while the others will provide extended release for the drug. Okay? Now, the third example for extended release اللي هو المايكرو encapsulation أو micro encapsulated drug. Okay, let me explain to you what happens. This is an example of micro encapsulation. Let me tell you what is happening exactly here, because this is from a liquid. Okay, so usually for micro encapsulation they use gelatin. This is a common one that will form will form the thin coat here that you see here, okay, around this droplet. You see here there are droplets, okay. This is the one that is forming the thin coat. But of course there are other materials that can be used to form the thin coat, like polyvinyl alcohol, ethyl cellulose, polyvinyl chloride. All these materials can be used. Like, what happens usually? First, you take, for example, gelatin, the one that will form the coat wall, okay, gelatin, and then you put it in a water, and then you add to it the drug, and you stir thoroughly, يعني strongly, so that the drug becomes very small. في solution the drug becomes very small, and after that you add another solution, okay, of another material called acacia. Acacia is like a gum. Now what this acacia will do, it will force the gelatin, okay. Now first the gelatin was spread all over the liquid, and the drug was spread all over the liquid. Now the gelatin will start forming a shell or a coat surrounding the drug. How? The acacia is forcing the gelatin to do that, to come out from the solution and form a coat around the drug. Okay. طيب, now this wall, okay, is a thin wall, and the layer, طبعا you will have a layer full of these droplets that you see here. Each droplet is surrounded by gelatin coat. You will have a concentrated layer of that. يعني في solution that you have, when you look at it, you will see that there is a thick layer, okay, containing these droplets, تجمعات منها, يعني gathering of these droplets, okay, as if your liquid is separated into two layers, one that has none or very little of these droplets, and another layer that has a lot of these droplets. Now what do? You take that layer, you separate that layer containing the droplets, okay, and then you dry them, and what you're gonna have? You're gonna have اللي هو dry, free-flowing, micro-encapsulated spheres. That's what you're gonna have, okay? Then the final dry microcapsules are free-flowing discrete particles of the coated material. طيب, الwall قديش نسبته؟ هذا thin wall that we talked about is from 2 to 20% of the total particle weight. هلا, the release depends on what? Drug release from this wall when it is administered. It depends on what? It depends on the ratio of the core to the wall. احنا حكينا هنا usually it's from 2 to 20. Of course, if it is more, the wall is more or thicker, معناته the release will be slower. The polymer used, is it readily soluble or not? And the method of microencapsulation. طبعا here we have only talked about one method, اللي هي co-acervation نحكي لها. The layer that I talked about here containing a concentration of these spheres are called co-acervate layer. Okay, هاي الدروبلتس بنحكي لها co-acervate. طيب, example number 4 شو هو؟ embedding a drug in slowly eroding or hydrophilic matrix system. شو يعني embedding؟ Embedding يعني mixing, putting the drug in slowly eroding. Eroding إيش؟ شو يعني eroding؟ You know, when you have erode, يعني when you have something like tablets, for example, and you swallow the tablet, or let me tell you a better example. When you have effervescent tablet and you put it in water, بتلاقوا the material at the surface starts to erode, goes out into the medium. هذا eroding. Okay, eroding is an تفكك or تكحط, removal from the surface. You erode, you remove things from the surface of the dosage form. Or hydrophilic, you know, loving water matrix. Isn't here the matrix system that we are using must be eroding or hydrophilic. Okay, these are the characteristics. One famous example for such matrix اللي هو ال HPMC. This is one commonly used matrix. طيب, what happens? How does this happen? Let me show you the figure here. Okay, you mix the drug with the matrix. Okay, يعني your drug, the circle one here you see, with the HPMC, you mix them together, you compress them, and you know HPMC loves water. So when you swallow it, when you swallow the dosage form, what will happen? the surface will be hydrated. Okay, so you have hydration of the surface. At the same time, the particle on the surface will start to get released. طبعا if the surface is eroding بصير عندنا ايش برضه؟ This will detach from the dosage form and will disperse into body fluid or GIT fluid. Okay. طيب, so what I said is the particle at the surface erode and dissolve, or مش erode, dissolves directly from the surface. But what happens to the particle inside? Now you will see here that you have a kind of gel layer or jelly layer. Okay. Now how this became jelly, the fluid, the GIT fluid got inside and made it jelly-like, and at the same time it dissolved the drug. So the drug will start diffusing يعني moving from the region of high concentration to the region of low concentration. إذن how the drug gets released? It gets released by tablet erosion and by diffusion. طيب, how do I control the release, make it faster or slower? By 1, the type of matrix. 2 is in the type of matrix, يعني HPMC or something else. Number 2, اللي هو by the concentration of the matrix. Okay, it's very important that once the hydrophilic matrix tablet is swallowed, it gels quickly at the surface. If it doesn't gel quickly, the tablet might break and you will lose extended release technology. Okay, so this is the embedding drug in slowly eroding hydrophilic matrix. Okay. Okay. طبعا we can compress it in tablets. Okay, but also we can put them in capsules. Okay. In case of capsules, if I want immediate effect, I can add some drug without the excipient. طيب. Now the same thing we described here, okay, the same thing, but instead of eroding or hydrophilic matrix, here I'm talking about inner plastic matrix. معناته الماتريكس is not dissolving here, it's not hydrophilic. Okay, so what happens here? The drug can only leave this matrix by not erosion, only by diffusion, because the matrix does not disperse, does not break, it remains intact. Okay, such as what matrix is available to us? We have polyethylene, we have polyvinyl acetate, we have polymethacrylate. We can use them to provide the inert plastic matrix. طيب, when you mix them with the drug and you compress them into tablets, the drug will be released slowly through diffusion. But what is going to happen? You know, this inert matrix will be excreted in the feces, in the stool, and they call it ghost شبح, ghost tablet. Okay, ghost. So you have to warn the patient that in the stool he might see a ghost tablet. It doesn't mean it did not give an effect or there is some problem. طيب, example number 6 is referred to as ion exchange resins. Ion exchange resins. Now, what happens in this way or technology to prepare an extended release tablets. You will have a cationic drug. Of course, you can have also an anionic drug, but this is an example. You have a cationic drug, and you prepare a complex of this drug with an ion exchange resin. Okay? طبعا resin is something that is not soluble. Okay? So if you have cationic drug, then you use an anionic or acidic resin. Why? Because you want to prepare a complex. What happens here? The cationic drug will go to the resin and it will replace the hydrogen, okay, for resin, and it will form a complex called resin drug complex. This can be washed and made into tablet, or maybe encapsulated or suspended in aqueous vehicle, whatever. Then you have this complex of resin and drug. So it can be of cationic drug and anionic resin, or anionic drug and cationic resin. Then when it goes to the GIT, whether the stomach or the intestine, a reaction will happen and the drug will be released from this resin. In order to know how this happens, let us see what happens in the stomach if we are talking about a cationic drug, okay, with acidic. So cationic drug, yani basic drug, basic with acidic resin. This is the complex. So when it's in the stomach, okay, شوفوا هنا, this something will happen. The drug, basic drug, will react with the HCl and will give me a salt. This is a salt called drug hydrochloride. It's a salt. Okay, so you have the drug released here. Type, and the resin will take the H from here and will turn into the acidic resin. طيب, now in the case we are talking about anionic drug, يعني acidic drug with basic resin, يعني cationic resin, see what will happen. Remember, here the resin is basic and the salt is the drug. Okay, so drug salt. Okay, and the drug salt is acidic. So what is going to happen? The drug will take H and will return to acidic drug, while the resin will take Cl and will come resin chloride. Okay, now the opposite will happen in the intestine. For the intestine, again, for basic drug, this is basic drug, acidic resin. Okay, see what is gonna happen. The drug, we said the drug is basic. It will react with Cl and form drug hydrochloride, and the resin will react with sodium and it will form sodium resin. Right, for acidic drug, what is gonna happen? Right, we said acidic drug. Okay, acidic drug, طبعا this is the drug, the salt. It will react with sodium, forming sodium salt of the drug, while the resin will react with Cl, forming resin chloride. Like, this is the way the release will happen. The problem اللي هي it's affected, يعني the drug release will be affected by the pH, not only the pH but also the electrolyte concentration. شوفي أملاح, what you are eating, the difference between individual and another, and this makes a problem. Okay, this creates a problem. because it means that the release can be variable. In order to prevent that, the polymer or a polymer barrier coating will be applied على إذن we will apply coating على اللي هو الion exchange resin. We apply coating. This coating will help to regulate the release of the drug, because the release of the drug can be variable depending on the pH and electrolyte concentration. طبعا I don't have to say that when the drug is basic, the release of the drug is greater in the acidity of the stomach. لما يكون ايش؟ the drug is basic drug. طيب example number 7, osmotic pump. Okay, osmotic pump, what is that? What I'm talking about? Here we are talking about system called OROS. شو اسم السيستم؟ OROS. OROS stands for O for osmotic controlled, R for release, O for oral, delivery system, O for oral delivery, S for system, OROS. Okay, طبعا this invention consists of a core tablet, يعني an inside tablet surrounded by semi-permeable membrane. شو يعني semi-permeable هون؟ It means it only allow water to come inside and outside. But what they do, they put in this coating, they put a hole. Okay, this hole is 0.4 millimeter in diameter, and this hole is created by a laser beam. Okay, let me show you here. So this is the semi-permeable membrane, only allow water in or out, and this hole will allow the drug solution only to go outside. But what do they put inside? They put an osmotic core. It's a polymer to provide an osmotic pressure so that it can draw water inside, and the drug is here. So when the fluid gets in, when the water gets in and start dissolving the polymeric osmotic material, it will create an osmotic pressure. يعني more water will start coming inside, and this will create a pressure so that the drug will be released in a controlled manner. Okay. طيب, what is this controlled manner? The controlled manner بيكون عندنا تقريبا release rate is constant, وعبارة عن few drops of water gets drawn into the tablet each hour. Okay, and this usually remains constant. Another thing that you should know, that what causes the release is the osmotic pressure difference from inside and outside. So the larger the osmotic pressure difference between inside and outside, the faster is the drug release. This is what you should know. Another thing you should know is that the release rate is also affected by the hole. What is the diameter of the hole? What the membrane we are using, and what's its thickness? All these factors will affect the release rate from the osmotic pump system. Okay. طيب, the inert component, remember, will remain undissolved and it will be eliminated in the feces as insoluble shell. معناته, what do you need to tell the patient? You need to tell the patient that you might see the shell in your stool. Don't be afraid, this is a ghost. Okay, a ghost shell. طيب, example منه اللي هو glibizide tablets. This glibizide tablet is used for ايش for diabetic patient type 2. اوكي اسمه البرودكت جلوكوتورول XL. طيب repeat action tablets, do you remember it? Repeat action tablets. Remember that we said that it usually contains 2 doses, right? One that is released immediately. It contains, sorry, 2 doses. Okay, it can be, as I said, a 2-layer tablet or a tablet within tablet, I told you. So the outer shell will be released immediately, or one of the layers will release the drug immediately, while the second will release the drug later, maybe after 4 to 6 hours of administration. Now, the inner core or one of the layers might be separated from the immediate release layer by a slowly permeable barrier coating, and this will allow the drug to be released over extended period of time. Example of that اللي هو repeatabs. Repeatabs you see it here, LaBayer. في عندنا البرودكت اللي هو كلارينيز nasal decongestant. Okay. طيب خلصنا. Now we finished the examples of extended release formulation or products. Now we will talk about اللي هو delayed release oral dosage form. Delayed. Now we are talking about ايش؟ delayed. طيب delayed معناته remember we talked about this, it's not prompt, it's not immediate, so it takes time and then the drug starts releasing. Usually we are talking here about releasing the drug in the intestine. What is the purpose of that? Why we want to leave it until later? Maybe to protect the drug. Maybe the drug is destroyed by gastric fluid. Or maybe to relieve gastric distress. Maybe the drug irritate our stomach and we don't want that to happen. Or maybe because it's better absorbed in the intestine. These types of tablets, we refer to them as enteric coated. Enteric coated. And they can be one time dependent. يعني time dependent معناته after 2 hours or 3 hours the drug will be released. طب وين راح يكون? Where will the tablet be? It will be in the intestine. It's like that, time dependent. يعني during this time the coating will ايش؟ slowly erode until it expose the tablet في الintestine. Or it might be enzyme dependent. Okay. Enzyme dependent يعني it is hydrolyzed by the catalytic enzymes في الintestine, or maybe it's pH dependent. Where is the pH dependent? Okay, or pH dependent. Here it's pH dependent. This is one. Is an 1 pH dependent, 2 time dependent, and 3 enzyme dependent. طبعا which one is the most common? اللي هو this is the most common اللي هو pH dependent, and you have studied that before. Among the agents used for coating: fats, fatty acids, waxes, shellac, cellulose acetate phthalate, if you remember. طيب, new types of tablets that can be fabricated as extended release. This is new types of tablets, okay, that are produced by 3D printing. Then it's produced by 3D printing. Okay. يعني مستخدم 3D printer. Yes, and we have already an FDA approved 3D printed tablet. Okay. This type uses layer by layer deposition of material من البرينتر using computer aided design. The reason why they are doing it, it's because there is shift in medicine design. في هناك now the design, we are trying to find a new ways to design tablets from manufacturing or industrial production of tablets or capsules. You know that they come with limited dose range. يعني they are not fabricated or they are not made to suit each patient. You know, we come with different age, with different weights, with different sex, with different يعني we have so many differences. But even though we have so many differences, we have the drug available in 1, 2, or 3 doses, not to suit each one of us. So the good thing about the 3D printing, it's tailored to the patient. يعني I can ask the printer to prepare a tablet for this particular patient who has this and this characteristics. Okay, يعني age, weight, sex, etc. Okay. The first example that was approved by FDA اللي هو سبريتام. سبريتام trade name موجود اسمه سبريتام, with technology 3D printing technology used called ZipDose. And the Spritam drug is levetiracetam, and this levetiracetam is used for epilepsy. It's used for epilepsy. It's fast dissolving tablet. You put it in your mouth and you just drink water. Okay, it dissolves very quickly in the mouth. طبعا the good thing about the 3D printer, it can print complex tablet geometries, يعني أشكال, different shapes of tablets that are not possible with conventional tableting machines. And we can use them to create, we can use the 3D printers to create modified release formulation. And for your information, our College of Pharmacy at Ajman University has a 3D printer, and we are currently working in that machine to produce tablets. Okay, this is the example I talked about, Spritam. طيب, what are the USP requirements? Okay, and guidance. Sure, requirements will guidance. First, about the drug release. Usually, once the company produces the tablets, okay, it has to do quality control test for the tablet, okay. And if you remember, one of the quality control tests, شو هو? Do you remember it? اللي هو dissolution. Okay, dissolution. Now, in dissolution, we usually take one withdrawal from the medium after, let's say, 30 minutes or 45 minutes and analyze that. هلا بالmodified release dosage form, it's not like that, and we have to refer to individual monograph. إذا لازم نروح للمونوغراف that إيش? The monograph that talks about that modified or extended release product and follow it. For example, and this is the type of apparatus is used here. For example, للأسبرين. If we come to the aspirin, the extended release aspirin, شوف ال USP requires what? It requires that you draw a sample at 1 hour, and the release should be 15 to 40%. At 2 hours, it should be 25 to 60%. At 4 hours, 35 to 75. At 8 hours, not less than 70. It's not 1 point. Okay? And this is only for aspirin. Now, for other products, you have to refer to the pharmacopoeia. For uniformity of dosage units, we have the weight variation test and content uniformity test. And they are applied, or they apply same as the normal tablets. إذن هنا زي normal tablets, the uniformity of dosage units, whether the weight variation اللي هو physical test or content uniformity اللي هو إذا this is physical test and this is chemical test. Okay, in vitro in vivo correlation. طبعا it's critical that when the company develop an extended release products is to establish اللي هو IVC, in vitro in vivo correlation. This will be very important for when you apply to FDA for marketing. It is important during product development, during clinical evaluation, during submission, even during post-approval when you want to make any changes in the formulation. Okay, you have to show IVC. طيب, what does it mean? شو يعني in vitro in vivo correlation? Let me explain it. In vitro is what happens outside the body. In vivo is what happens inside the body. So you need to establish that, for example, if 20% of the drug dissolve in 1 hour, what is going to happen in vivo? مثلا, let's say 10%, for example, will dissolve or will be absorbed or will be available to systemic circulation. So you have to do kind of correlation, how they are correlated to each other. Okay, if they are not correlated, this is very bad. Of course, we know that the better the dissolution, the better is the absorption and bioavailability. The FDA provide guidance how to establish or how to develop, if you have a product, a new extended release product, how to develop in vitro in vivo correlation and how to see whether it's able to do prediction. يعني من in vitro I predict what happens in vivo, in a human. Okay? Also it provide guidance to establish the solution specification اللي هو ايش؟ The one I talked about in aspirin. So in the pharmacopoeia they will write, okay, depending on the IVC, they will write these things. Is then to establish the solution specification. Also, if later after marketing you want to make a change, okay, you don't need to experiment in a human. You can use the IVC, correlation between in vitro and in vivo, to get as a surrogate, يعني بدل, instead of doing bioequivalent study on human. So you can use the IVC prediction. Okay, and this will reduce a lot of cost and trials on human. طيب, when you want to establish اللي هو ال IVC, okay, in human you need to use usually from 6 to 36 human subjects in order to establish correlation. Okay, is an for bioequivalence, in order to establish correlation you need to use from 6 to 36 human. And the type of study we use is referred to as crossover. Crossover, when you are using those human and you are using two formulation, مثلا A formulation and B, okay, and maybe C or D or whatever. So the same person will take both product with a washout period between taking the two products. يعني, for example, the first day he will try product A, and then after 1 week he will go and try B. Then he will take one dose here and one dose here. This is at day 1, but this one is at day 7. So there is a lot of time in between. So no carry out or no carry out effect. That is wash out period. بنظف حاله. So what I mean to say is same person is taking drug, the two products, okay, when doing comparison. طيب, this is preferred طبعا, this is preferred. This is preferred. But sometimes it might be acceptable to do parallel studies. معناته لا, here some subjects will take product A, and some human subjects will take product B, and then we do the comparison. وفي عندنا cross study analysis. معناته no, I'm gonna take only, I will try one product, okay, with my human subjects, and then I will compare it to another study, okay, using the product. بس ايش هذا البرودكت؟ عبارة عن intravenous solution. لأنه الانترافينوس solution ما في complication. You directly put the drug in the body system, or aqueous oral solution because its absorption is very fast and has no problem, or immediate release dosage form that is absorbed very quickly. Okay, that is called cross study analysis. So you need to be aware of these different types of study. Labeling, what you should be labeling. طبعا in addition to usual labeling requirements, you need to indicate, for example, whether your tablets are enteric coated or not. For example, United States Pharmacopoeia بتحكي الاسبرين delayed release must be stated في الليبل انه this is enteric coated. Theophylline must be stated it is extended release. But in addition to that, it has to say that extended release every 12 hours or 24 hours, which one, and which in vitro drug release test apply كمان. Okay, يعني it should state extended release 12 hours or for 24, and which in vitro release test should apply, because there are 7 different tests available. Okay. طب شو هي the last thing we want to talk about? What are the clinical considerations in the use of oral modified release dosage form? What you should tell the patient when you give them this new extended release dosage form. لازم تنبههم. You should let them be aware, and you should remind them that this is extended release. It's not like the normal one. So they have to take the medication, for example, once a day, not 3 times a day. So you have to bring that to their attention. You should ask them not to switch their medication with another modified release formulation. يكون عندهم modified وفي company ثاني عندهم modified, then they switch. No, they cannot, because each one will have different release feature. So they should not switch their extended release product with another one, or with an immediate release product. Also ask them not to crush it or chew it, because this will affect the modified release product. It will destroy the coatings. Also ask them not to use the product as a source for drug, maybe for example to prepare an oral liquid for pediatrics or for children or babies. It should not be used for that. Okay, it has modified release ingredients, and it should not be used as a source to prepare other dosage form. Another thing, or the last thing, اللي هي if they are taking, for example, osmotic tablets or non-erodible plastic inert plastic matrix, you should tell them that they may see a ghost or empty shells in the stool. So warn them about that. They should not be scared of this. Okay, I hope everything is clear. If you have any question, please do not hesitate to ask me. Yalla salam.